Perfect. So thank you all for coming today. This is another one of our meetings where we are trying to help promote physician scientists' work among early stage investigators who are in neonatology. We conducted a survey of people who are early stage investigators in neonatology about the topics that they thought would be helpful to learn more about over the course of a year or a couple of years. And this is the next topic that was most popular from that survey, which is how to actually, realistically start a research program and build a research lab. Today, I just want to thank Patrick Myers, who encouraged us to put on this series of panel talks or discussions. I also want to introduce and acknowledge my co-moderators for this series, who are Betsy Crouch at UCSF. Do you want to just say hi, Betsy?
Hi, everyone. Thanks for having me.
And Misty Good at UNC.
Hello, good afternoon. I'm excited to be here.
I don't think I said my name. I'm David McCulley. I'm a neonatologist and physician scientist at UCSD. And I'm really excited to introduce our panel. I tried to pick a group of people who I thought would be really informative to this group, who are at pretty early stages of their career or who recently moved. And I thought that would just give really realistic advice for people who are either still in training or considering building a research program, to give like real nuts and bolts tips about how to do this. So I am really excited to introduce our panelists today. If you guys, I'm going to introduce you one by one, but if you could talk just a little bit about a couple of things. If you could say what stage of your career you're in, what your area of research is, the type of work you do, like what actually goes on in your lab, like do you work with cells, do you work with animal models, like that kind of thing, just to give people an idea of what your environment is like. And then what is your actual lab environment like? Is it a wet lab? Is it a computational lab? A little bit of both, like that kind of thing. So the first person I want to introduce is Krithika Lingappan. She is a lung biologist who I've gotten to know really well over the last couple of years. She is a neonatologist at CHOP and just recently moved her lab from Baylor to CHOP. Krithika, can you just talk a little bit more about your situation?
Yes, thanks, Dave. Thank Misty and Liz for having me on this panel. So I'm going to answer the questions that Dave wanted me to answer. So our lab is interested in figuring out the mechanisms behind sex-specific differences in neonatal morbidities, the organ of interest being the lung at this point of time. I moved from Baylor College of Medicine to CHOP 2 years ago in 2021. It still feels like yesterday. I have not fully recovered from it. So my point of joining today was actually also to learn what to do right. I have done a lot of things wrong, so I can also tell you about my mistakes a little bit in this transition. So looking forward to this panel. Thank you, Dave.
Great, thank you. Can you just talk a little bit? So what do you work with? Like, what's your lab and yeah.
Yes, so our lab is a basic science lab. We work with mouse models. We also work with primary cells as well as cell lines. We also have in vitro systems built in our lab to study the effects of hyperoxia and hypoxia on those cellular systems. And I think we are recently, I would say last year and a half, we are also, I would like to say, a computational lab, trying to do some translational as well as single cell studies and multiomics in our lab as well.
Perfect. Okay, next I'd like to introduce Jill Chang. Jill, I got to meet a couple of years ago when she was moving from Northwestern. She is now at the University of Colorado in Denver, and she is a neuroscientist and neonatologist there. Jill, can you just tell us a little bit more about yourself?
Yeah, thanks, David, and everybody else. So yeah, I moved to the University of Colorado about a year and a half ago. I had done my training and then stayed on as junior faculty for 4.5 years at Northwestern before coming here. So I'm 6 years out from fellowship. So I guess technically still considered early career investigator. And what I do is I use a mouse model of placental insufficiency to study how intrauterine growth restriction leads to white matter injury and the cerebral palsy phenotype. And so I mostly do basic science research using the mouse model, although I'm venturing into more cell culture now as well. And then as far as my lab environment, the one that I've moved into, it's actually pretty unique here at the University of Colorado. And it was one of the things that drew me here. It was the community feel to it. So the setup is there is a separate facility where there are 11 investigators from the section of neonatology, and we are housed at this facility. 4 are PhDs, and then the remainder of us are neonatologists, physician scientists. And so there is a good community here that I thought at my stage of training would be very helpful when starting up my own laboratory. And just for context, I moved with a K08, so I still have that mentored grant.
Perfect. Thanks, Jill. Last, I'm excited to introduce Jackie LaJeunesse, and I just want to acknowledge and thank Lila Nolan, who introduced us. I was looking for somebody because I had really good contacts in lung biology and in neuroscience, but I wanted to have a slightly broader panel, and Lila suggested Jackie. So Jackie is a neonatologist and a physician scientist. She has an MD PhD and is at Indiana University. She's an immunologist and at an early stage of her career. So Jackie, can you just tell us a little bit about your program as well?
Sure. Thanks so much, David. And thanks, Lila, as well as Misty and Betsy for putting this on. So I am a very new investigator. So I just graduated from fellowship in July. So I am working on getting my lab set up here at Indiana University, which is where I did my fellowship training. And I'm interested in preterm birth. And so I work primarily with mouse models of preterm birth and looking at kind of the maternal immune contributions to that. So I work on mouse models, I work with primary cell lines, and I'm mostly focused on how the role of T regulatory cells and maternal tolerance during pregnancy and how that can change the changes in the in utero environment can affect prematurity as well as postnatal phenotypes for the offspring.
Perfect. Thanks, Jackie. Okay, so our main objective today is to try to talk a little bit about how to start a research lab, because I think a lot of people kind of have in mind that they're interested in doing research, but it's one of those things, and Jackie and I were talking about this yesterday, that we get specific training in a lot of things, but we don't get specific training in many more things that we end up encountering over the course of our career. And I think starting a research lab is one of those things where you're just kind of like, well, I think that might happen. I don't have any idea how to actually do it. So I thought it would be helpful just to kind of go around this panel and talk a little bit about our experiences. And then Betsy, Misty, and I can also add in because we, recently started research programs or moved research programs. So I think we can probably add to the discussion a little bit. So just in the beginning, it's mainly about how did you physically set up your lab? That could be a little bit about like what the environment was like, what you needed to purchase, how did you manage and think about purchasing things versus hiring somebody, who gave you advice about that, and that kind of thing. I know this is an incredibly vague question, but Krithika, do you feel like you could help start that off just by talking about like your initial transition to starting your lab at Baylor, and then differences when you moved your lab from Baylor to CHOP?
Yeah, let's start writing that book of what not to do.
Yeah.
So when I started, I started like Jackie. So I had not gotten my K yet. I was hired as an assistant professor. I wanted to work on this area of sex-specific differences, but knew nothing about being a researcher. So kind of made it up as I went along, I guess. I was I did not know how to negotiate for a startup, did not know what to ask for a startup, did not know that you had to negotiate for space. So none of that. So I think it's as blind as you can start blind, I guess. In my startup, there was no money to hire anybody for the first 2.5 years or 3 years. And after that, when there was money to hire someone, I think there was just enough money to hire a technician. So at that point, and I had to bring this in the perspective of the fact that I had not done research before, in the sense that I had not done any extensive amount of research during before I became a fellow at Baylor College of Medicine. I also did not do schooling in this country. So there is no undergraduate study period in India. You go from high school to being in med school. So there was also not much of experience there. And my mentor was a toxicologist. He was not a lung biologist. So it was like the perfect storm of not knowing what to do. So the very first person I hired was a research technician, not because I wanted to hire a research technician, but because that's what I had the budget for. And then there was a lot of trial and error there about what kind of research technician should you hire? Should you have a fresh out of college person? Should you have a little bit more experienced person? The pros and cons of that. So there was trial and error there. And then the third person stuck with me for 2.5 years. And during that time, I was successful in transitioning to a K. And then I just wanted to say at the outset that, and I want to limit myself on time to tell me if I go beyond 5 minutes. I don't want to talk for more than 5 minutes. I have always had a small lab. So in Baylor from 2012 to 2021, when I moved, the 9 years, I only had a lab that had 3 people, including myself. So it was me, a senior research tech, and a research associate. I was very afraid of having a big lab because I did not think that I would be able to manage all the data, and 2, I was too paranoid of not generating data or looking over the data by myself. So I was afraid that if there were too many people in the lab, I wouldn't know what's happening in the lab. And of course, the other big reason was never had the money to have more people than that. So it was a lab of 3 people. And then when I moved from Baylor to CHOP. So when you move as a mid-career researcher, and this is when I had 2 R01s, and I was moving at the midpoint of the 2 R01s from one institution to another. The institute invested, in me during that move. So there is a startup that was provided to me, but I made the really bad, wrong, not thought through decision of having this weird bravado within me that I could move labs with nobody moving with me. So those 2 people that were with me in the lab were rooted in Houston, could not move with me. But I somehow told myself that, yeah, so what? I did it like 9 years ago. I can do it again. So I came to CHOP with no one. And the hiring process took 6 months to hire the first person. So I felt like I was back, so I bred all my mice again, had to open all the labs, set up the lab, order stuff, etc. So I know we'll get to the details there. But in terms of guidance, I think the guidance that I had were senior investigators here within the division who provided me guidance on how to hire and how to hire in this institution. Because the other thing that I learned was each institution is so different, pros and cons, limitations, time frame, money required, etc., that it was a learning process for me all over again. So I'll stop there, but I'm sure there'll be more questions down the road.
That's perfect. I feel like I can echo a lot of those experiences. One thing that I think was helpful for me was I had a really good advocate when I moved after fellowship from UCSF to University of Wisconsin in Madison, who was the division chief in Madison, Deanne Pillars, who encouraged me to put together a budget for a startup, which I felt like I wasn't sure that was what I was ready for, but remember physically going around the lab and looking to see like, what do I actually use every single day that I would, really need on a bench top? And if I had a couple benches, what would I need? And that was the basis of, my budget. And then I went around and saw like, what is the bigger equipment that I use more regularly? What are the large item expenses that I have less frequently, but are still there that I should put on the budget? And how do I, budget for someone's salary? Jill, can you talk a little bit about just as you were kind of building your program in Northwestern, and then when you moved to Colorado, how that went for you?
Yeah, I mean, a lot of the themes that Krithika mentioned and trials and tribulations, I was like, oh, I am living that life and have lived that life. So I'm hoping that a lot of the people in the audience can hopefully learn from us. But similarly, so when I started basic science research, really in fellowship, became serious about it. And as I mentioned, I stayed on as faculty. But I was not able to get really much money at all for a startup. And I also did not know how to negotiate that. What I got was only enough to do experiments for about 1 year, and not, to hire any personnel. I also did not get very much protected time, which, as we know, is also really important, especially if you can't have somebody else in the lab to help you out. And so those are two things that I feel like you should really advocate for and are necessary. I did have some lab space in my mentor's lab, so I had that, and there was some shared equipment, so I didn't have to worry about buying like large, expensive equipment to use. But it was, so for the first 4 years while I was at Northwestern, I really was just doing the experiments on my own and just, having to fund myself through the grants, and thankfully being able to obtain a KO8, which provide you with funds to do experiments, but still not enough to hire any assistants. So that's something that I think is really important to think about when you are graduating fellowship and you're pursuing this career, is to make sure that you can negotiate for things, because I think that perhaps my path would have been easier if I had some of those things moving along. And then having that knowledge, when it came time for family reasons, for other reasons, but mostly for career reasons, wanting to make sure that I put myself in a good spot to continue my research career, I made sure to, when I interviewed at different places, and once I had, more leverage to negotiate and knew what to ask for, I made sure to ask those questions about startup funding, lab space, community and mentorship, which I think have all been very crucial in me moving and setting up a lab. And that's those are the large things that helped me choose University of Colorado to come to. And then since coming here, I've experienced now that I have funds from my startup to be able to hire a research technician. It definitely is, there's a whole art to that we definitely are not taught. And I am now on my my second hire. First one, I didn't quite understand what I was looking for or what I needed. And now I have my second hire, and I think hopefully we're in a good place now, but that is another topic too.
I was just
Perfect.
gonna add onto something that Jill said, because I think it's really important, is like when you're moving or you're trying to figure out, maybe you're not moving, you're launching your lab from, the institution that you're at, you may be entering another space, for example, and may not just be like right next to your mentor. But I think it is really important to, whatever space you're going into, to know your space, what is around you, what are the resources. And if you're going to a new institution, not just like what you are going to buy, but also, what cores are there that you could use, especially Krithika mentioned, like, it takes a long time to hire people. And so especially if your science is going like ongoing, you can utilize those cores until you get your own, lab teammates to help continue on your science and outsource a little bit until you're able to get, you know, through that rough phase of hiring everyone. So
And that's definitely how I survived at the beginning, was getting these pilot grants through the cores that could help me get that preliminary data to keep moving on, and to help finish some of those research, some of that research that I couldn't get to myself.
agree, because you don't want to have to buy all these expensive pieces of equipment if like there's one on another floor or the same floor or right around the corner. So it's important to ask all those questions. So it's good that you brought that up.
Just
Yeah, I
to
think-
offer another strategy, David, I'm really impressed with how you like walked around and wrote down the equipment and how much it costs. I mean, maybe I'm lazy. I just asked a friend who had recently gone on the job market for her list of, what she asked for a startup package, and then modified it so it looked like it was mine.
That's
So
what
yeah, I mean, I think that's really impressive, David. But I might recommend just like not rewriting, not what's it called, reinventing the wheel. So
Yes.
just grab somebody else's package and then make it, update it for you. So anybody, like any of the 6 of us, would be happy to send you. Like I have a version from a couple years ago, but you could update it.
Yes, I'm happy to share that as well. It was really good. And I should say that I have a very fortunate position. My wife, my partner, is a neuroscientist, and we were moving together from UCSF to Madison. And she put together a much more elaborate budget than I did. And hers was the, skeleton. So it helped me just in terms of like thinking about how to organize it, but then realistically, like what were the things that I needed? I did literally go around the lab and, it was just helpful for me to think about like, what do I actually really need? But the truth of the matter was that I think good advice is to just be patient because of the point that Missy made. It takes a little bit to see what the new institution will be like. And even if you're staying at the same institution, some things that you may be able to share become evident over time. And you just don't feel like you need to build a lab and have like all the pipettes and all the equipment and everything set up in the beginning. Like that, I don't think is usually how a really efficient lab gets started. It happens over time, and it's okay to be really patient about that. Jackie, can you talk a little bit about that? Because you're right in this transition. You have a little bit different background than Jill or Krithika in the sense you did an MD PhD and probably knew a little bit more like what you were going to build your research program around. But can you just talk about your experience?
Yeah, so I will say that we get a little bit more of kind of didactics or education surrounding kind of how to start up a lab. And I had already kind of known I was going to do the physician scientist track for a long time. So there was a little bit more understanding of some of those steps. So I was really fortunate to have great support from both my chair here, who had been one of my mentors throughout my training, as well as my division chief, who both of them are physician scientists. So that was actually really helpful to have people from the very top kind of already advocating for me and having kind of things in mind for what I would need for a startup. So that was something that kind of I didn't have to work super hard for or push very hard for. That was kind of part of the agreement for kind of recruiting me here, which was great. So I had a startup funds. I had a spot on an institutional K-12 right when I started up with I'm working on submitting my own K-08 now. But those things were kind of established. I kind of had talked with my mentor. I have space in my mentor's lab. So just like Jill was saying, like, I don't have to make all of those big purchases right away. And my startup funds were kind of structured so much so that I have a significant amount of money up front for everything. But then once I get my K and have my own space, there's an additional kind of influx of money that will happen at that time to kind of buy some of those bigger equipment that I might need. So really kind of initially starting off, I'm still looking to make my first hire. So I'm like literally next week I'm sitting down and talking with HR and putting out the ad. So any advice you guys have, I'm open. But most of my initial purchases have been kind of like, you know, mouse cost, gel electrophoresis, those types of things that I use on the day-to-day, kind of like David was mentioning in terms of like, I did that. I kind of like walked around and I was like thinking about like, okay, what do I actually have to buy? Like I need one for myself at my bench type of thing when I start. So those are kind of the things that I've been doing right now.
Perfect. Thank you so much. Betsy or Misty, are there other things you would add to that just based on your own experience?
I was going to say, I have somewhere a document on how to hire the best people that somebody else made. So I'll offer that. I don't know if she wanted me to share it widely, but if you want to email me, I'm happy to email it to anybody who wants it.
Yeah, I
Sounds
think it's
good. Yep.
important to hire the right people, right? Because you want to make sure that it takes so much work to like get rid of people. So you want to be sure. I would just add for, especially since Jackie, you're hiring, call the references yourself. I can't tell you how much time I wish I would have, you know, when I've hired the wrong person. I have let, you know, HR folks like talk to those people or, you know, get a survey or something like that. I call them yourself. Like this is your lab family. And when you have somebody that's out of not, you know, not performing, it brings the whole team down. So even if it's one person in a very large team. So my advice is call them yourself, definitely.
I have a
Okay.
two-step interview process that I used even for my technicians, where the first interview was an interview, you know, where like I asked standard questions. For the second interview, I had them read a paper and then we discussed it together. And that was kind of a test as to whether we could talk science. And that was very informative, I'll say, where there were candidates that it became very clear that we weren't, I don't know if they didn't understand my science or they didn't know where I was coming from or they weren't paying attention or anyway, it was a good weed out. And I think it actually was an example of it's sort of like how coders have to code, you know, they have to do a task during their interviews. This was like an example of somebody actually, you know, performing science in the way that I expected them to perform on a daily basis during an interview. So I found that very helpful. I have friends who for postdoc candidates will ask them to put together a mock specific aims page on a project that they'd like to work on as an interview. So I've started doing a two-step interview process for all of my hires. For the grad students, the first step is like I'll let most people rotate in my lab, and the second step of the interview is their rotation. So we haven't talked a lot about grad school affiliations, but that's what I've done for that.
I think that's a good point that Betsy brought up. I also do two-step interviews. And so I usually interview virtually first, just to myself to kind of weed people out. And then I have them meet with various members of my lab, either in, you know, in person or virtually. That way there's multiple people interviewing them and seeing how they perform in different environments with different levels of comfort with various folks. So
I do exactly the same thing. I have two steps and I have just one-on-one interaction with them in the first step. And then the second step is they come into lab and meet with everybody. And I don't have that many people, so it's easy to do that. But that really is informative about how they will interact with the other people in your lab, but also show them my work, kind of like give a talk, but then ask questions along the way about like what they do. You know, I work a lot with mice. I want to see like have they handled mice? Do they set up genetic crosses? Like, do they know about the like genetic tools that we use? Like that kind of stuff. Krithika brought up a point that was similar to what was a major concern for me when I was thinking about balancing hiring versus acquiring equipment, which was just around, you know, how do you pay for these people? Because in addition to the, you know, thinking about how to manage incoming data, a big barrier for me was just thinking like, am I going to be able to realistically pay for someone's salary for, you know, more than a few years? Like, am I going to be putting them at risk where I'm going to have to, you know, say that I can't afford them anymore? And I was just wondering, as you were kind of getting yourself established, did you have a fund manager that you were meeting with that was helping you think about your budget, and also like when you were thinking about paying for some big ticket item versus paying for someone's salary, did you have either a researcher who was helping you think about that, or did you actually have like an administrative fund manager who was helping you think about that? Can you answer that, Kritika?
Dave, as you were asking that question, I was thinking that I probably went through an emotional roller coaster, like, during this transition. Like on some days I'd be like, oh, I can do this on my own. I don't need to ask anybody. I've done this for 9 years. I mean, why do I need to ask anybody? And then on some days I'd be like, I don't know anything about anybody, and I just don't know what to do anymore. Maybe I should just quit and look for something else. So there were phases like that. And then on the day that I would feel super confident, I would make and on that confident day, if I decided to buy a piece of equipment, I bought a piece of equipment. So one of those buys that I did when I transitioned to CHOP was the Leica microscope, which was I think $160,000. But there wasn't a microscope around me that I saw that I could use, and I needed one, and I needed a good one. And I think the startup gives you this feeling of impunity that, oh, I can buy like 1 or 2 pieces of equipment on my own that I didn't have before. And again, I think those were, I could have rethought some of those purchases, but I did. And then on days that I was not sure what to do, I would, beg and reach out for support about, either hiring people or equipment for that matter. And I think for me, one of the things that was eye-opening was the mice colony management costs also differ so much from one place to another. So I think I was paying 2/3 of the cost that I'm paying right now at CHOP. So I think your budgeting can be thrown off even by that, by how much like your mouse colony management is going to cost from one place to another. I think the way I operated when I transitioned was I bought the pieces of equipment that I knew that I had to have, without which I could not do research. So that meant a PCR machine, a Western blot imager, a microscope, couple of centrifuges, a water bath, et cetera, et cetera. But then I decided to focus on people. I don't think there can ever be like an investigator who has a K without divisional or departmental support. I don't think they can hire or keep personnel for any reasonable amount of time, given the uncertainty of funding. They talk about daily wagers, people who live on a day-to-day. I think like a researcher, I feel like that, like every 3 years, you're like in a crisis mode, like, oh my God, am I going to keep everybody in my lab? Do I need to start let people go? Who am I thinking about letting go? And who's where in the hierarchy of the lab, etc. So I think you toy with these questions on a constant basis. It just never goes away. At least for me, it has not gone away. So I think that question of who, how many, how long, I make sure that I am very transparent about the uncertainty of hires in my lab, because I tell them that I'm a researcher. You're going to be paid by a grant. The grant starts on this date and ends on this date. My goal is to keep you in the lab. My goal is to relentlessly keep trying for funding, but I'm a researcher and your salary is contingent upon me being funded. Most of the people who hire for positions like that know how research positions work. What do they call it? Like when the position is being funded by hard money versus soft money. So yes, if the position is being supported by a division or department, that would be great. I have one such person in the lab, but rest of all the people are funded by my grant right now. So I think you just have to be transparent that's how it works. But the success of a lab also depends on the group that's in the lab. So I want to fully back up what Misty said, especially when you're transitioning. I think It's first who, then what. Especially if you're thinking about charting a further course for your lab, I think you have to be very, very diligent about who you're bringing on to the team before you chart out the further course of your lab.
One thing I'll just add to what Krithika said is, especially when you're on a K and you don't have much money and you may or may not have startup, as people mentioned, you can be creative about your hires. So thinking about, would you be able to support a pre-med in your lab, either for free if they're willing to volunteer, or pay them a small amount of money. They would be cheaper than a technician. You would probably get them up to 20 hours a week. They would be, you'd have to teach them, obviously. So it'd be a little more intensive than, say, somebody with experience, but and certainly you get what you pay for and what you're willing to support. But when you think about all the great pre-meds that are out there, and certainly we were all pre-meds at one point, having a great mentor and learning about science and having somebody support them is really important. So for us, like, we usually have 4 to 5 pre-meds in the lab at any given time, and I do pay them because I think it's important, and helps for them to show up, et cetera. But I do think you can be creative in that way. Medical students, certainly residents are a little bit harder unless they're PSTP residents, but even clinical fellows, do need great mentorship and need a project that, they can work on what it is that you're doing already.
Can you talk a little bit more about that, Misty? I don't know if anybody else is in this situation. I have like an undergrad type person who emails me once a day who wants to come volunteer in my lab.
Sure.
And what I've learned, I guess, is that the ones who have had previous lab experience, or it's just, I think I have to be careful about the other people in the lab, because it can be a lot. I picked a couple of ones that were not great fits in the beginning, who actually weren't that serious about wanting to be in the lab. And it was a big bummer for the grad students who were training them, who then felt like their time wasn't valuable. And that grad student, I need to be productive. So I have some ideas about how to pick better undergrads, or, you know what I mean? Folks who aren't going to be in the lab full time, let's say, as like a general category. How do you select them?
I go through the same process in terms of interview. So I interview them. I have them interview with multiple members of the lab. I have a little bit bigger lab now, like anywhere from like 15 to 17 people, depending on the number of undergrads that we have. So they interview with at least 4 people. I have them come to a lab meeting to see how they interact, so they know, that they'll be presenting, and make sure that they're actually available on lab days. I will say that I have had some bad undergrads, and when I was at WashU, did have to kind of bounce a couple out of the lab. So I do put them on like a semester by semester basis. And so basically everyone that's in the lab has to say like whether or not they can continue on. And if, one of my lab manager or one of the PhDs or, anybody else in the group says like, they're not really coming, or they're a little bit of a problem, then they're out. And so I try, especially with pre-meds, because they can be very competitive. I try to have like just 1 per year, so they're not like, trying to compete for the same med school spots or, things like that. But I really just tell them up front, like, you can't be competitive with other people. Like, I need you to show up. Otherwise, like, we have people that are waiting to get into the lab. When I first got here to UNC, like, I was starting from scratch, like Krithika mentioned. I didn't move anybody with me from WashU, although I did have people that I had interviewed that I was going to bring at WashU and said, hey, would you come to UNC instead? And so that worked out for me for 2 of the people in my lab. But I will say that when you're doing that, when you're starting from scratch, or you move to a new place, I just reached out to the pre-med advisors and said, this is what my lab is like. If you have any great pre-meds, like, please send them my way. And for me to get my first undergrad here, like it worked really well. And then after that, like they just blasted some listserv. And so, and then there was like 25 people that reached out in like 2 hours. So then it was just like going through the interview process. So you can get a lot of people with free advertising that way in terms of undergrads. But if they're reaching out and you're, like you said, Betsy, if they're reaching out, there may be some interest that they have in your work. So then just try to tease out what it is, because sometimes it's a personal connection that they have to your work. And I find that those folks are more motivated. Like if they have an interest in prematurity, or if they had, a sibling with, necrotizing enterocolitis, or, something like that, they tend to work harder.
I agree with that. I think those people with those compelling stories are the ones who are really motivated. I have to say I've had the best luck with just word of mouth recommendations, and I've asked people in the lab, like if you have friends who'd be interested in working here next year or things like that, that has worked really well. And my first hire, who was somebody who had left a PhD program, but was moving to Wisconsin at that time, and had really a lot of the same experience that we really wanted to have in the lab, worked out extremely well. And that was only through word of mouth, but um, it's, it's just tricky. Jill, can you talk a little bit about just personnel and the two questions? One is just like, do you have a fund manager who helps you manage your funds and thinking about hiring people? And now that you've moved, like how is it in Colorado and managing like personnel in your program? I think you're muted. Sorry.
Muted. Yeah, there is a fund manager who does give quarterly, and then if I ask for it, more frequent reports of my financial fund or where I am in my funds. But I think, I just kind of want to circle back to, hiring people, because again, this was my first experience ever doing so. I think it's really important. And I, of course, was keeping an eye on my funds and thought, oh, I should get an entry level because that costs less money and I'll be able to support somebody for a longer period of time. But I think what's really important is to think about where you are and what you need at the time that you're hiring somebody. So even though I wanted to be more fiscally responsible and go for that entry level, which is what I initially did, what I came to learn is at this time point, with setting up a new lab, coming to a new institution, I just didn't have the bandwidth to invest in as far as training and teaching somebody at the bench that I may have had more of a luxury to do at my prior institution, where I knew the lay of the land. I knew the clinical, weren't as many things that were taking my time. And so that was one of my first pitfall with hiring, because I guess I wasn't realistic about the type of support that I needed. So I think that's really important to think about. And, I have always been gung-ho as far as teaching and mentoring. And so I thought, well, I can teach, I can mentor. I love doing that, but I just couldn't do that at that point. And so I think that the second time around, I hired some more of a mid-level technician, and that has made all of the difference, because this individual is able to be more independent, which is really helpful at this stage for me. So I think that's something important to think about. Also, because in my environment, although I have other people at the Perinatal Research Center, other PIs who have their research technicians around, they do different things than I do. So there's not other people who are able to help train somebody new to this bench. So take that into consideration as well. Like if you're not there to answer questions, or if there are not other people available, if you're not in a large lab, that I think is really important to think about when hiring as well. But yeah, as funds, I'm still learning that, because this is the first time I've had more extensive funds to be able to manage. So I'm still navigating that right now. And it's still on a small scale with just me keeping a track of it. But I think I'm going to have to rely more on others to kind of help me manage the funds.
Thanks, Jill. I was talking about this with Jackie yesterday, and I think this is a good point to bring this up, but I had a institutional KL2 award that helped me get established in Madison, and that was helpful for funding reasons, also protecting my time and stuff. But it also connected me with a peer group in Madison who were not all in pediatrics and certainly weren't neonatologists, but they were all physician scientists and were trying to, establish their research program. And it helped for a bunch of reasons. We had monthly talks that addressed this kind of thing, like building a research lab, like hiring people, mentoring people, establishing, effective mentoring relationships with people who are your mentors, and then becoming a mentor yourself. And also just gave us plenty of time to like, discuss and kind of commiserate about these things. Jackie, I don't know if you can talk a little bit about that, like what your peer group is like in Indiana, or do you feel like that is a group that you can ask these kinds of questions with, is helping you as you're trying to hire somebody now? Or do you have a network of people who are outside of your institution that you are connected with?
Yeah, so I think it's incredibly important to have a peer group, whether that's within your institution or outside. For me, I've been asking some of like the new kind of PhD hires, some of like the questions about just hiring and like how do you reach out to the cores and like get all that stuff kind of situated. But within neonatology, we kind of have a group of like physician scientists that meet once a month to kind of go over kind of research ideas. And so I'll kind of pick their brains individually. And then being on I'm on an institutional K-12 with one other physician scientist. So we kind of help each other. And then I have a couple friends who went through like the MD PhD program with me that are still at the institution that are just a couple years ahead of me. And I often will reach out and ask them because sometimes you have kind of very high level mentors who've been very successful and been doing this for forever, and they don't remember what it was like when they were first starting up. So it's been really helpful to kind of have the big picture goals based on those mentors that are much more established, and then be able to talk kind of nitty gritty details with people who are a little bit closer to it. So that's been very helpful for me.
And you and I were talking about this yesterday, that in addition to like the, just how do you run a lab, there's also some things that seem like a huge burden that you've just not encountered before. Like, how do you write an animal protocol? How do you write a biosafety protocol? How do you write a job posting? A lot of those things you can just get from somebody who is a year or two ahead of you, who hopefully has just gone through this themselves. And then you don't have to modify it much. You at least have an idea of like what you need to submit. And then, as you and I were talking about, it's not something that has to be like the best perfect thing that's completely done when you turn it in. It's sort of like you're turning in what you think is a reasonable thing, and then you can have an interaction with whoever the administrator is who, is going to help you navigate getting it validated or certified or approved or whatever. So I feel like having that network of people at your institution is really helpful for that, but then also having a group of people who you're kind of just sharing this experience with outside your institution is just really supportive as well. I want to leave some time for people to be able to ask questions from the audience. While you are welcome to put questions in the chat. If you would like, you can raise your hand and we can call on you. Or we can just keep going with this discussion. I want to say that in an effort to support this idea of networking among early stage neonatology physician scientists, Jonathan Knowlton and Ben Feinstrom are going to have a discussion right after this meeting about how to maintain kind of an ongoing stream of discussion for people who are early stage neonatology physician scientists. And the idea is to have a slightly less structured ongoing discussion about that. So stay a little bit later after this meeting is finished to talk about that. But please feel free to ask some questions. If we don't have anything, I would be curious just to ask the panel and just kind of go one by one, with Krithika being first. If there is one thing that you have learned that you wish you knew as you were trying to start your lab, what is that one thing? Or if there's one, thing that really went wrong that you learned from that you think this group would benefit from knowing about, what is that? Kind of a hard
That's a
question.
very difficult question.
Yeah.
But okay, since you had asked for questions from the audience, I don't have a question, but I had a request.
Oh.
I knew about Jill's and Jackie's research because they're both on this call. I did not know they existed. So, and I don't know how many of other neonatology physician scientists that I don't know about. I was wondering if everybody who's on the call today can just put in the chat like what they work on. Like I'm a neonatologist
great.
and I do XXX just because I'm curious. I want to know what all of you do. I might not even know. So anyway, so that was like my one request. So what would I not do? I think maybe I would not be as unaware and naive as I was when I started out on this research journey. I think if I was better informed for my own good when I started out as a young assistant professor, I think I would have done a better job advocating for myself. But because I went through what I did, I am super passionate about not letting another young person make the mistakes that I did. So I tell them that how important it is to advocate for you when you start or when you transition, because those are the times when your demands will be catered to. Once you're there, kind of forgotten until something major happens. So negotiate when you can, negotiate hard, negotiate like you're the only person in the world, negotiate like you are the only one who matters. Don't worry about anybody else. So I think I would tell that to my younger self, I guess.
Yeah, if you don't get a no, you haven't asked for enough. I think that's a good rule of thumb.
That is really good advice. Fully support that idea. I will totally say that with a lot of imposter syndrome in my head all the time, I have a really hard time doing that. But if you need a pep talk going into that negotiating meeting, like ask for it. Seek out the mentor who is the strongest advocate for you and just say, like, I really need, like, is this a reasonable thing to ask for? Tell me what I should do. And then like say what you think the barrier is, like, what am I, how should I handle this? So get some advice going into that meeting. Just get a pep talk of like, you are worth it. Jill, do you have one thing you wish you knew or something that didn't go well that you want to share?
I think the biggest thing was also advocate for yourself. And yeah, I'm introverted and huge imposter syndrome and thought that, I can't, if they say no, like maybe it's because that's the way it is, or I'm not worth it, or things like that. No, you need to advocate for yourself to be successful. And ideally the times are when you're starting or transitioning. Those are the times that you will be most successful, I feel like, and have the most leverage. And it's okay to say, okay, they're not valuing me. I need to go someplace else, because that was also a lesson where I stayed longer than I think I should have, and I should have advocated for myself and looked for other opportunities sooner. So I think that's really important. And then also creating a community, like a scientific community of mentors and of peers, as you've heard from this, is incredibly important. And it is something that I struggle with, because I would love, as a physician and a scientist, I feel like I definitely like to be by myself and not talk that much, but it is so important to make those connections. So I think that's also something to try to push yourself early on to do that as well.
Perfect. Thank you, Jill. And Jackie, do you have something you'd like to share?
I echo a lot of what has already been said. And one of the things that David brought up for me was I was so worried about like doing some of the administrative stuff that I didn't have great, very specific instructions on. I wanted to do it right. And I was so worried about making a mistake or looking stupid because I didn't know how to do it right, that it took me longer to submit it. And so it pushes things back. And so I've finally gotten to the point where I was like, oh, okay. No one actually knows exactly how to do this, and they just submit it and get feedback and then fix it and send it back in. So kind of just getting over that barrier in my head to be like, it's okay to not do it perfect and go from there.
Yeah, I feel like, what is it? Perfection is the enemy of the good or something like that. Like I struggle with that all the time, but that is really good advice. Um, that is great. Betsy or Misty, do you guys have anything you would like to share? Because I know we talk about this stuff all the time. Am really excited to say, if you haven't been able to catch it, Betsy, Misty, and I have been trying to support neonatal physician scientists in several ways. And one of the ways that we have been lucky to be able to do that recently was through a collaboration with the Incubator podcast. And we just had our first program come out last week where we got to interview Betsy and hear about her career path and things like that. Related to her work, trying to understand the mechanisms behind germinal matrix hemorrhage. So if you get a chance, check that out. We'll try to have programs that are coming out monthly. And as Betsy said, as we were starting this program, we will probably look to interview many of you. Our goal is to interview neonatal physician scientists, hear about your exciting work, and try to support this community in any way that we can. We're kind of rare people, and we feel like being able to connect with people around the country is a great way to support one another. Do you guys have anything you'd like to add, Betsy or Misty?
Yeah, I was just gonna say, like, this is a great community and we want to continue to have it grow. So please tell your friends, and this will be recorded and Patrick's gonna put it on somewhere where he I don't know if he's there, but he's gonna upload it. So if you're in the lab working and you weren't able to hear the whole thing and you want to catch it again or share it with your friends, you should be able to. So that's great.
Thanks,
Oh,
Krithika.
DVD.
Yeah.
Oh, DVD YouTube
I will
channel.
say, yeah, Ben and Daphne from the Incubator podcast wanted to make sure that we had proper equipment. And my voice sounds a lot cooler with this microphone. So that's my tip.
I was
for
going
all
to
of
say,
you.
your voice
Um,
sounds like a rock star.
Oh,
Did you change your voice after you got the equipment? You're like, I need to make my voice deeper and more like a podcaster voice.
no, not at all. I try not to think about my voice at all. So
You have a great voice.
I have I guess an announcement and then a random tip. And an announcement is that I'll stay on for a bit to talk about a new opportunity for TCAN members to co-author review articles with a WECAN, which is the more senior neonatology group. There were a couple of them that are in the works, and Stephen Perlman, who I think is the WECAN chair, identified mentors to partner with TCAN members. And I think that's a great opportunity to, stretch your writing skills and also establish a connection with somebody who's, has a lot of expertise in neonatology. If I'm looking particularly at all of the folks who don't have their video on, which is totally fine, but we're looking for volunteers to write those articles. So cool opportunity. Okay, my tip. I started using ChatGPT for different things in the lab. I'll say that a lot of people, all of the coders in my lab, which everybody is a coder except for me, I'll say, which is an interesting situation to be in. And if you are younger than me, please learn how to code. That's something I would do differently if I could go back. It was different a decade ago. But I have started to use ChatGPT. And ChatGPT has become my new tool to write a letter recommendation. I don't totally use ChatGPT for letters, but it's a great start. It just gets you over that hump of like, oh, I've got to write this letter. So you feed the ChatGPT the person's CV and they'll generate an outline. And then I'll go back and write stuff I really care about, rewrite stuff that I really care about. But that's streamlined that process a lot. So there's my random tip for today about how to decrease your workload for all of the things that we're expected to do as neonatal physician scientists.
Perfect. Betsy or Misty, do you want to add one last thing? And then I'll say thanks.
No, I mean, I love ChatGPT as well for letters of recommendation, especially when people like provide a draft and then you can, have it rewritten and then edit, which is easier than like the blank page. So I second that. The only thing I would say is we talked a little bit about finances, and I put it in the chat, but I'll say it out loud. I think when you're first starting out and you have a little bit of grant funding, like knowing where it goes, because you can, easily spend it and you're like, oh, I have money until you don't. And so that happened to me when I was on my K. It was like, oh, your K is spent. And you're like, oh no. So I would it also happened to me with my first R. So we spent it within like 7, 8 months instead of the year that it was it was supposed to last. So I'll just say make sure you keep track of that, or make sure you meet regularly with somebody who's keeping track of it, whether that's a research um grant administrator or something. Definitely schedule regular meetings with them, because it'll help keep you on track. And then if you do have multiple grants, making sure that you spend on them, at different intervals, so that you're not trying, the last month, you're like trying to spend as much money as possible and stocking up on, supplies. So I would just, from a financial resource standpoint, keep that all in mind. And we're all available if you need to reach out to us. Please just shoot us an email. We're happy to answer any questions and help you through it all.
Thank you, Misty. Okay. Betsy asked us just a quick turn on your camera so we can take a quick photo to post this event. And I really appreciate you doing that, or if you're comfortable, hopefully. I just want to say thank you to, I don't know if someone can grab a photo. Let's see. To our panelists, thank you, Krithika, Jill, and Jackie, for taking time for this. Thank our audience for coming. Hopefully that was interesting. Thank you, Patrick, again for encouraging us to put on these sessions. And thanks to my co-moderators, Misty and Betsy. I really appreciate you guys helping me with this. I will take a quick screenshot. Boom. And thank you guys so much for attending today. So stay on if you would like to talk a little bit more about how to maintain kind of an ongoing communication for neonatology physician scientists who are early stage career investigators. I think I will hand this meeting over to John and Ben, who are going to run that session in the meeting. So if you're not interested in that, feel free to go ahead and jump off now. And I really appreciate you attending today, and we'll have another posting for our next session probably in the next couple of months. So thank you all for attending. John and Ben, this meeting is yours.
Thank you, David, so much. This is awesome. All you guys, thank you all so much for sharing your wisdom with us. Really